Bea Marin-Diaz, and she is currently a postdoctoral researcher at the University of Florida's Engineering School in the Center of Coastal Solutions with Dr. Christine Angelini and Andrew Altieri. She has a master's in ecology and ecosystem management and restoration from University of Barcelona in Spain, and her PhD is in ecosystem-based coastal protection from the Royal Netherlands Institute for Sea Research and the University of Groningen. Her professional interests are in seagrass meadows, marshes, and oyster reefs, and nature-based solutions. And she is working on a really exciting project that is funded by the Department of Defense REPI Challenge Program and Restore Bay County, called Enhancing Tyndall Air Force Base Coastal Resilience Through Nature-Based Solutions. And that is also a collaboration with the Nature Conservancy, Jacobs Engineering, and the U.S. Naval Research Laboratory. So a really exciting background on that Bea has with private sector and government and ecology and university science. And so it's a perfect interdisciplinary talk for our Nature Coast Currents today. So with that, I will pass it over to you to start sharing your slides.
Thank you for the introduction. Okay. Sorry, my voice is not the best today. So today we'll present part of the work that Savannah introduced, and this is the one about seagrass restoration in Bay County. The title is Exploring Feedbacks Preventing Seagrass Colonization by Using Passive Restoration Techniques. And this work is part of the East Bay Living Shoreline and Seagrass project led by the Bay County from Florida, and is in collaboration with the Nature Conservancy, Jacobs Engineering, and the University of Florida. The goal of this project is to restore 5 acres of seagrass to increase the resiliency of Bay County's coast. Our role, so the role of the University of Florida in this project, has been to identify the processes preventing seagrass colonization and also inform the planting design and test passive restoration techniques. I will give more information now about this. So this is the project site. Here we can see Florida, the Gulf of Mexico, and we are in the northwest Florida in San Andrew Bay. If we zoom in a bit more, here we find again the Gulf of Mexico, but the field site is inside San Andrews Bay, East Bay. So there is also the West Bay, but this is East Bay. This field location is next to the Tyndall Air Force Base, and the aim is to restore seagrass to improve the coastal resiliency. Like seagrass provides many benefits. It reduces erosion of the soil, it can attenuate waves, it provides a lot of habitat, and also traps carbon, among other benefits. So if we zoom in the area and we look in the history of the area, the oldest photo we can find is from 1953. So it's like more than 70 years ago. And by then it was already bare. We don't know the exact reason why, but it's the same time that the Air Force Base was being constructed. So maybe there was some sedimentation happening. But the thing is that it has been closing. You can see the red line is the lastest delineation of these bare areas. So it has been closing over time, but it hasn't closed completely. So we wonder if there was a big sedimentation event, and then it has been, like the seagrass has been trying to close, but not completely. So we start from this. If we look into the seagrass that is present, we find Thalassia more in the deeper areas, and then in the darker areas that are shallower, we find Halodule and also a bit of Rupia. And Rupia is especially found in the shallowest areas mixed with marsh vegetation. And then all these areas are bare. So I will bring you now a bit under the water to see what we find. This is what you see when the seagrass starts to become bare. So it's kind of transitions from dense seagrass to bare sediment without vegetation. And we found a lot of stingrays. The stingrays to feed, they vary, like they dig pits to eat animals from the seabed, and they create a lot of bioturbation. And in this area, there is a lot of bioturbation by stingrays. You can see it even from Google Maps. All these little points are feeding pits. The image on the right is just incredible, the amount of feeding pits that there are. So yeah, in summary, there is a lot of bioturbation. There are also ghost shrimps, which are shrimps that dig holes in the sand. Horseshoe crabs that you can see in the photo, when they move, they also move the sediment around. And also the two photos below are stingray pits, and you can see how they can uproot seagrass. In contrast, when we swim around the meadow with seagrass, we don't find as many ghost shrimps or any ray pits at all. We did some surveys of ray pits in different patches to check that this is happening in more areas, not only in this small study area. And we did surveys in bare sand, then in the edge between seagrass and bare, and then inside the meadow. And as we can see, there are more ray pits in the bare patches and in the edge with the seagrass than inside the meadow. The biggest pit that we found was 1 meter wide, like diameter, and 30 centimeters deep. And in this photo, you can see me trying to sit inside the pit because it was huge. And on the left, you can see some of the more common stingrays. This one is like about 30 centimeters diameter, so not as big, but there are all kind of sizes. We also did a preliminary survey of ghost shrimps in the bare area and in the seagrass meadow, and we can see that in the bare and in the seagrass edge, the ghost shrimps burrows are in very high densities. This means that there are a lot of shrimps there, and inside the seagrass meadow, they are in very low densities. So this brings us to the hypothesis of this study, that is that we have a bare sediment. In this case, we don't know for sure why it became bare, but it can happen from a blowout of a boat, or naturally during a storm, or if there is sediment deposition from the waves. Like, it can be natural or it can be also made by humans. But the thing is that ghost shrimps like bare sediment because they can dig the holes easier than inside the seagrass meadows. And in addition, the stingrays like to eat ghost shrimps. So this can create this feedback of, or we hypothesize that this can create this feedback of, we have bare sediment, the shrimps prefer this sediment, the stingrays go to this area because there are more shrimps and they start to bioturbate. So the new seagrass that could occupy this space can't, because it's being uprooted by the stingrays. And this could maintain a loop, of leaving this area bare. So to examine this a bit further, we did 3 studies. They were kind of done at the same time, but I will go first with the stingrays. So one was, can we exclude the stingrays and help the recolonization of seagrass? Another study that we did is what is happening in the sediment, like, are there sediment changes in the elevation? Is there burial in the area? And then, this is a spoiler, but there was some burial, so we analyzed that also. So starting with the stingrays, we installed 2 types of exclosures, and then we also had control plots. We did this in 2 patches. So we had treatment control, which was only 1 bamboo marking the plot. Then we had a treatment that was a perimeter with bamboos around in a square, and then a grid, which were bamboos around the perimeter of the square of the plot and also inside. And we did this in the seagrass edge and in the bare sand in 2 of the patches. So along these blue lines, we install the grid perimeter and also control plots. This is how they look in the field. The control is just one bamboo, and then the perimeter, as I said, this one was installed in the edge of the seagrass, and then the grid. And the response variable that we measured was grid cell count, because counting shoots of this small seagrass was not feasible. So we just counted how many cells had seagrass before adding the treatments and after. So the results were positive at the beginning. The disclosures work, they exclude stingrays, and also there were floating fragments of seagrass that could establish inside the disclosure and started growing. To see a bit more the data, I will guide through this plot. The yellow is the control plot, so no bamboos, and then the green and blue are the plots with bamboos for the ones that were in the bare sand and the ones that were in the seagrass edge. So after 1 month, we can see that there was more seagrass in the disclosures than in the control. So there was seagrass growing. Then in September, after 2 months, the same, there was seagrass growing. Then during winter in February, it was not as clear. And after that spring, there was a general retreat in the area, not only in the disclosure, but there was a lot of algae growing, and this kind of caused like anoxia conditions, and some seagrass was retreating. So after that, we could not really measure properly the disclosures. But the thing is that at the beginning, the seagrass could establish. However, over time, we also saw that the seagrass was being buried. And also during winter, it dies off a bit. This is the seagrass that we could find. So these fragments were initially floating in the water, and then they fell in within the disclosure, and then they started rooting there. So they made roots. So now we will see why they were being buried. This is the part where we were analyzing the sediment dynamics. And for this, we tried different things. One was to put seagrass mimics, which were these cores that you can see in this image in the right. And we put them inside in the plots in the bare sand, in the plots in the seagrass edge, and also 1 meter inside the meadow. And the results were very clear. After 3 months, the mimics inside the meadow, so where there aren't stingrays or shrimps, were still there, but the ones in the seagrass edge and in the bare sand were buried, so were covered in sand. This is a photo of how I could find them. So basically, I had to move a bit the sediment, and by moving the sediment, I could also find a lot of buried dead leaves of seagrass. And here you kind of see the fake seagrass that now it's black because it was decomposing. But there was a lot of sediment on top. Then the other thing that we tried were the sedimentation plates. This is a technique that was developed by Bell many years ago. And it's basically a plate that you dig below the ground, and it has a string so you can find it back. So when we are in the field, we insert a metal rod, and we insert the metal rod until we touch the plate, and then you measure the distance from the plate to the sediment surface. And if you do this over time, you can see changes in sediment elevation. So we install these plates in the disclosure plots, both in the bare sand and in the seagrass edge, but also inside the dense meadow, which are the green points. And we also took RTK elevation. So this is like real elevation in the field in July 2022, and then after 1 year in July 2023. So the first results of the plates show that there was high sediment accretion in the bare sand and the seagrass edge. This looks like a lot, but everything is showing the same. Each color is 1 month. So it goes from the green to the orange. And each plot is the bare in the patch 1, bare plot in the patch 2, and then the plots in the seagrass edge of the patch 1 and the plots on the seagrass edge on the patch 2. All of them show that there was an increase of sediment over time. The maximum arrives to even 20 centimeters of sediment accretion. So this was suspicious already, because if every year there is like sediment to like 20 centimeters of sediment deposited, it would be kind of an island, no? Like there isn't that much sediment, so that every year there can be this much accretion. So we look at the RTK, the real elevation of the ground, and what we found is that the plates in the seagrass edge and in the bare sand were actually sinking. They were being buried. And here we have some data that prove this. The gray boxes is how much sediment was deposited, measured with the sedimentation plates. And then the black boxes show the actual elevation change measured with the RTK DGPS, so the real elevation change. And we can see that the plates showed a lot of accretion, but the elevation remained more or less stable. There was some variation, but because also the ground is not super flat, so there is always a bit of error. And in contrast, the plates that were installed inside the meadow were stable. They didn't show accretion. So this means that they were not sinking, because there aren't rims varying and messing around, the plates were actually in place. So after seeing this, we tried one last thing, which was to install bamboos with burlap fabric to try to exclude the shrimps. So we put in the field these squares of burlap. Burlap is a biodegradable fabric, so it's not like you don't need to put plastics there. And then because we couldn't put a sedimentation plate because you could break you have the fabric, we track the sedimentation with skewers and also seagrass mimics like the ones I showed before. So again, this at the beginning was looking okay. The burlap was excluding the shrimps, but it also sinked over time. So we think that it's because the burlap was too small. So all the bioturbation around the burlap and with the natural flow of the water was being deposited on top of the burlap as well. So in conclusion, passive restoration in this situation was not enough. We found an area with dense meadow without ghost, well, not without, but very few densities of ghost streams and rapids, low sediment turnover, and low seagrass burial. But then you have these bare areas that are full of rapids, full of ghost streams. All these animals create sediment turnover and sediment burial and seagrass burial. And when we try to exclude the stingrays using bamboos, it works and it traps seagrass initially, but over time there is burial, likely because of the shrimps. And also the natural recruitment was too low to, like you need a minimum percentage of seagrass to survive over time. And the natural recruitment by the floating fragments was too low that they would not survive it was not enough quantity of seagrass to overcome stressors. Like if you would put here a thick plot of seagrass, maybe it would survive because the more seagrass, the lesser shrimps, they exclude each other. And then if we, in addition, put burlap to exclude the shrimps, we also saw that there was boreal. So probably because the small plot size. So how do we translate this to decisions for the project? One option was to consider the area as not suitable for restoration. It could be leave as a stingray and shrimp habitat and other benthos that live there. Also, especially because we didn't exactly know why it was there. If you know that it was because of a human impact, even if there are now stingrays and shrimps, you could think, no, we have to restore it. But if you don't know the origin, maybe it's just natural. And if you really need to restore it, that's a whole new discussion. The sediment turnover must be addressed, and maybe you would need transplants to have a minimum seagrass quantity to make it to survive. Doing some literature review, other options that I have seen to exclude the shrimps, which would be the main thing here to address, are pesticides, which can be from the ethical point of view, and if it can spread to the adjacent habitat, it's a bit, problematic. The foundation, which is putting a plastic on top of the sand to create anoxia, and this basically kills everything. So it's like pesticides, but very localized because you only kill the animals below the plastic. Then the burlap that we used has been used also in other experiments, but it can be ripped off and sometimes it doesn't stay in place. And another option is high density planting, but like for high density planting, you need a lot of donor material, which is very expensive, and also you destroy other habitats. So there isn't any ideal solution yet to address this ghost shrimp issue, but that could be something to investigate further in the future. That's it. I would like to thank all the people that have been with me in the field over these 2 years. And thank you for your attention. If you have any questions.
Excellent. Yes, you do have a couple questions. So the first is, if there is net accretion, isn't it essential to identify the source of the sediment in order to prevent continuing accretion?
Yeah, so the net sediment accretion was found with the sedimentation plates, but then we saw that the sedimentation plates were sinking. So it was not that there was more sediment, more new sediment on top of the plates, but because they were sinking, we were measuring every time like bigger depths, but because they were sinking, not because there was new sediment on top. Also, the creek may bring some sediment, but not to these quantities.
So it's just the same sediments moving around within the bare patch, you think?
Yeah, it's like the shrimps. I don't have the graphic here, but there is a study that shows how the shrimps create these very deep burrows, like more than 1 meter deep, and they eject sediment like a volcano. So they get sediment from the bottom, and then they eject it like a volcano. So if you have something like a plate and they grab sediment from the bottom and eject it on top like a volcano, that's the process we think it's happening here.
Yeah. A follow-up question to that is, alternatively, will continuing accretion be a long-term positive by supporting future establishment of salt marsh in the face of sea level rise? So I
Yeah,
think you kind of answered it, but if you want to comment on that as well.
yeah, I mean, if there was real net accretion, which we haven't seen, yeah, but we don't think it's like new sediment coming. Also, we like some questions that I had before is because one of these patches is directly connected to a creek, is the creek that it's delivering more sediment. But in the surveys that we did, we also monitor some patches that were not connected to creeks or anything. And they were also bare and full of rapids and shrimps.
Next question is, what is the scientific name of the ghost shrimp you were looking at?
These ones are Calinaceae, the shrimps. I have the shrimps in in the freezer to identify the specific species. But yeah, the family name is Calinaceae. It's not like the typical shrimp that we can imagine, pink with the antenna. These ones live below the ground. Like you would not find them swimming. You have to dig in the ground to find them.
Yeah, how deep are their burrows?
I have read from literature that more than 1 meter.
Wow.
In this site, we could not test. It's very difficult to analyze this. The studies that I have read, they put some like cement or something similar to fill burrows and then extract the shape.
Yeah.
But we couldn't do that.
All right. Well, there might be a couple more. Oh, here we go. Are the shrimp everywhere, or do they like certain types of seagrass sediments?
We found shrimps in all the patches in around that area, in shallow sediments. Like I also surveyed more in the sandbars that are a bit more offshore, and there were some, but in less density. So the higher densities were in these shallower areas close to the marsh. And this water is less saline than in the Gulf. So in the Gulf side, I have another location and I can't find the shrimps. So it's more kind of bay waters, I would say. In this site,
This side is more salty.
this site is less salty
Oh, less
than the
salty.
Gulf. Yeah.
And you mentioned briefly that the edge was retreating. Have you continued to observe the edge retreating, or has it since stabilized?
Here the seagrass edge has been closing. It's the only thing I have is the ones from the photo that I showed at the beginning. So it kind of stabilized and is not closing faster or anything.
Okay.
I had a question that you kind of alluded to already, but how common are these shrimp and seagrass meadows and this interaction with the with the rays? How common is that in other places? Like is right around Tyndall, like is that a really unique thing, or do you think that's happening in less saline bays across the Gulf?
Yeah, I saw them also in the West Bay and also in Sunnyvale, but I have read studies that they are also in the Caribbean. There are different species in the Caribbean. I think they are bigger and they make big ejection cones, like really big, that you can see them. They are very apparent. These ones, you only see the opening of the hole. So that's why at the beginning, we didn't think of them being the problem. Well, the problem in quotes, like, because in the tropics, you see the big cone and you say, okay, they are really bioturbating, but here it was less evident. And then in the West Coast, I think in Oregon, they also have these shrimps competing with seagrass. So yeah, they are quite common.
All right, well, if you have any last questions, get them in quick because it looks like we don't have many more. And so I want to say thank you again so much for coming and presenting at Nature Coast Currents. And we do have one more webinar in this year's series before we take a little break for the summer. Our next one will be Abby Scrow, who will be talking about scallops and some of the microbiome and pathogens and parasites of scallops just in time for scallop season. So I'll send a follow-up to everybody with a link to register for that, as well as the recording for today. So thank you all so much for attending, and thanks again to you, Bea, for being our speaker.
Thank you.
Goodbye, everybody.